FORGENIUS is a project devoted to the characterization of the European network of conservation of forest genetic resources that is organized by the FORGEN program. And it runs between 2021 and 2025, and it will end up producing a new information system that will contain information on forests, European forests, both from the point of view of ecology and ecophysiology and genetics, plus the modeling of their resilience to climate change. So I think that without any further ado, I can, maybe that there's a microphone that is open, maybe we should close it. I don't know who is, thank you. So I can give the floor to Nassim and we can start. Thank you very much.
Thank you, Ivan. Hello, everybody. Just a few seconds to share my screen. Can you see my presentation?
Yes, we can.
Okay, good, thank you. So, I'm very happy and glad to kickstart this second edition of our FORGENIUS public webinars. So, I'm Nassim Belmokhtar. I'm from Biopharma Research Unit at INRAE of Lyon in France. I'm also part of the INRAE Phenobois platform dedicated to physicochemical characterization of trees and woody products. So today I will talk about the use of near infrared spectroscopy for high throughput characterization of European forest genetic conservation units. I will start by giving a short overview of our research approach in this project before describing the near infrared spectroscopy methods and technology. And I will give you today a couple of examples of developed models to date. And I will finish by, I hope, some interesting perspectives. So in this FORGENIUS project, I'm involved in the WP3, and this work package is dedicated to provide dynamic descriptors of genetic conservation units by integrating genotypes, phenotypes, and environmental characteristics as indicated in this chart. And the ultimate goal of this approach is to evaluate and forecast population adaptive capacity. I'm happy to work in this work package with numerous partners from different countries across Europe. And here in this map, just to show you how we decided to sample trees in order to provide the phenotypic trait for the near infrared spectroscopy. So we identified 15 GCUs in different countries. And from each GCU, we have 10 trees sampled to provide a data set of 150 samples in order to develop this trait calibration. Once developed, the models will be applied on larger data set on one GCUs. On 2 GCUs, which makes 1,000 trees to be predicted for each species. So I will now talk about the near infrared spectroscopy, which is based on the use of the electromagnetic light ranging from 800 to 2,500 nanometers. This range of the electromagnetic light is characterized by very strong energy, and this light is used for our research in order to estimate the reflectance of samples at numerous wavelengths. So the near infrared light will interact with tissues or material, and will target the molecules vibration motion. And this vibration motion will give us absorbance at each wavelength, and these wavelengths will give us this kind of spectra. So this reflectance is, of course, depending on the chemical composition of the material studied. We are studying. So we use this technology because it's fast and reliable analytic technique, and also because it requires minimal or non-existent sample preparation. We can analyze samples directly without any previous preparation. This kind of spectral data can provide physical and chemical information of virtually any kind of matrix. And by combining this data with multivariate analysis methods, we can perform quantitative and qualitative analysis. But this method is known to be not suitable for trace analysis. And it's also known for the complexity of the spectra due to the presence, to the overlapping of absorbance band. We have the same information repeated at different parts of the spectrum. So this makes less straightforward spectral interpretation in contrast of HPLC or other chemical or spectroscopic analytic methods. And of course, to be able to work with the NIRS, it's mandatory to perform chemometric methods. So let's now talk about the models we developed in this project. I will present you today models developed on Populus nigra and Pinus pinaster. But before that, just a few words about our experimental approach for the spectra acquisition. So we work on two kinds of samples, on short cores of 5 centimeters and short branches. In both cases, we will collect spectra on solid and powder samples. So the solid is used for minimal sample preparation and for faster prediction later when the model is set up. When we work on powder, indeed we have the grinding step to do before the measurement, but we will get much more molecular information, which is sometimes needed to the prediction of hard, very difficult traits. So in our case, we used the benchtop FTNIR spectrometer, ranging from 1,000 to 2,500 nanometers. We scan our samples 64 times to get an average spectrum representative of the analyzed material. The second step of our experimental approach concerns the model's development. So after spectral acquisition, we have to prepare the data for the calibration. The first step will be the pre-treatment of the spectral data, because it's known that even, as I said before, we know that the reflectance is mainly related to the chemical composition. that is the chemical bonds in the material, but we also have some noise related to the temperature, moisture, and other parameters. So we have to apply some pretreatments such as normalization, derivation, detrending, smoothing, and so on, in order to extract the useful information for our use. And after that, we move to the exploratory analysis using principal component analysis, where we will study the clustering. We'll observe the clustering of the samples based on different factors, such as countries, GCUs, and other known factors in the data set. This step will also allow us to identify any outliers in the data set, which must be removed before moving to the calibration. And for the calibration step, we start by splitting our data set when we have a large data set. We can split it randomly or using other methods into calibration and validation data set. In this project, we focused on the use of PLS regression method combined to cross-validation in order to identify the best model. You can see in this graph different colors corresponding to the different pretreatment applied to the raw spectra. So the best model identified will be the model showing the lowest error of prediction during cross-validation. And after identifying this model, we'll apply it on the validation dataset validation dataset. And we have some guidelines to select our model. We have to ensure that the error of prediction is as low as possible. And also we have other parameters such as R-squared and RPD, which must as high as possible. We have many other parameters that can help us to decide which model is, if the model is accurate or no. So here comes our models. So the first model for previous graph will be so we try to predict many traits, but the best models obtained were with P50 and P12. I present today the P50 model. So we can see that the results here were obtained using based on short course ground spectra. And the model we obtained showed R-squared of 0.89 and an error of prediction of 0.08, which is low compared to the range of variability in the measured dataset. So this model was used to predict two whole GCUs, the Spanish and the Australian one. And the results of these predictions are shown here. We can see that the range of variability is almost the same with compared to the measured ones. Those results were used by Aida Soleil from India in Spain. And she gives interesting results when focusing on how much of the variation of this trait is due to variation in genetic factors. So she calculated the irritability, and the results were quite good. I move now to the Venus-Penastel models, and here we developed the models based ground branches, because we got only ground branches in the beginning of the project. So we can see in this graph that we have variable responses according to studied traits. We have good results ranging from R-squared ranging from 0.1 to 0.97, and good models for P50, P12, SLA, LMA, and GMIN. but sometimes we struggle to get something interesting even for wood density. We don't know why, but that's our results obtained with ground branches. So here we showed the result of some models, P15, SLA, and GMIN. We plotted the observed against predicted samples. So the same approach is currently applied on sawdust provided by Amelia Gonzalez Araujo from CityMass in Spain, in order to predict the traits, the validated models on the wool GCUs. And as presented before, we will also calculate irritability for those traits to see if there is any relation between genotype and the predicted phenotype. So after presenting those models, what next? What can we do now? So we are now pushing up to predict validated traits on Pinus pilaster, as I just said before, on wool GCUs. And we will also deliver models on other species before the end of this year. We are working currently on Pinus sylvestris and Fagus sylvatica using the same approach presented today. And at the end of this analysis, we will try to set up a new multi-species approach to see if it can help us to bring something new, or if it can improve our models, for example. But today we are also looking to trying to further explore our NIRS data by using it as a new trait or a genetic marker in order to indirectly capture on the phenotypic variants to compute relationship matrices for predicting complex traits, to replace costly and time-consuming methods by near infrared spectroscopy, to capture some genetic variability by estimating the irritability of absorbances along the spectrum, and also maybe for mapping corresponding QTLs. So this is what we call today phenomic selection, which is radically different from the classical approach I presented today. The advantages of this phenomic selection are the possibility of the use of NIRS as regressors at the genotypic level rather than the individual level. This will allow making prediction at any environment, because I remember that near infrared spectroscopic data are very sensitive to the environment and the prognosis, for example. Then that's it for me, and I'm now ready to answer to your questions if you need to know.
Thank you very much, Nassim. We are slightly behind schedule, so there may be time for a couple of short questions. I don't know if I'll manage to see raised hands, but
No, let's see.
You can just speak up if you have questions. But I just have a short comment. Of course, I think this is a very, very promising approach and successful as we see it today, in enabling us to get estimates of traits that are very, very complicated to measure and very sometimes expensive. So I think this is really a way to go to generalize the assessment of functional and ecological properties of those three species. So thank you very much, Nassim. There's a question. I think there was but I don't. There's a hand raised. Nicola, please.
Yes, thank you, Ivan. Thank you, Nassim. So you probably said it and I missed it. The NIRS was measured on different tissue, right? So for when you estimated gemin, it was on leaf that you measured the NIRS spectra. All right?
No. I only get in this project we only measured the spectra on for Pinus pinaster on branches.
Okay, so
So
even with the spectra on branches,
yes,
you can predict the gemin of the
yeah,
leaves.
it was really surprising to be honest. But yeah, the model said that the correlation between the measured gemin and the spectra collected on ground branches was easy to make.
Excellent. No, that's really nice. Yeah, thank you. Very, very impressive work. And amazing. Thank you.
Okay, thanks. I think we will hand the floor back to Nassim to introduce our invited speaker, Matthew Robson. I'm very pleased that you could come and present. Thank you very much. Please, Nassim.
Thank you. So I'm really pleased to give the floor to Matthew Robson, Associate Professor and Program Lead for Woodland Ecology and Conservation, coming from the University of Cumbria. He will be talking about the integrated phenotyping of ecophysiological traits for comprehensive fitness assessment. So the floor is yours, Matthew.
Okay, good morning. Thank you very much for the invitation to speak with you today. I'm very happy to be able to present a little bit of my research and also talk a little bit, giving a broad overview of what I see as being interesting and important with respect to your FORGENIUS project as well, which I only know a little bit about, but we will see. So I am an ecophysiologist, ecologist. I've been working at the University of Helsinki, so I will present some of the work of my research group there, and now I'm working at the University of Cumbria. I also am a member and lead author on the UNEP panel looking at the environmental effects of ozone depletion and climate change together. So that gives some of the broader perspectives that I'll talk about as I go through my talk today. So I mainly work with light, but I'm going to try to talk about light and climate change together and think about the context of how we see changes in the forest affecting plant growth and what this means when we try to link genotypic traits with phenotypic traits. So in particular, we work in provenance trials and we work in controlled conditions. And sometimes we don't always see the same effects that we expect in controlled conditions and when we look at traits in forests or in provenance trials. So one of the things that I'll try to understand first of all is whether there's a reason for this and what the reason might be that we don't always see the same across the two types of environment. And then I'll go on to talk a little bit about potential changes in the light environment that we might see and how plants might respond to that, and new technologies available that might allow us to do ecophysiology and phenotyping a little bit more rapidly with a higher throughput, so that we can really get to identify how to link phenotypic and genotypic traits and the mechanisms that drive these changes in the phenotype that we see. So first of all, perhaps to talk about how we see trade-offs amongst traits in nature. And we give you this classical point of view from Sandra Diaz from Argentina, collecting a whole range of traits across plant species and seeing these fundamental trade-offs between productivity and stress resilience, resistance to change, between plants that are more conservative and plants that are more exploitative in their strategies, seeing differences in leaf morphology, leaf physiology, differences in height, differences in seed mass, depending on where plants are able to allocate their resources and whether they're optimizing their growth for production and competition, or whether they're optimizing their growth for stress resilience. And this is something that we can find, for instance, in species ranges and what we expect to see across species ranges. So here we have a range of silver birch, Betula pendula, across Europe, and we see a lot of small populations isolated in the southern range edge, and our expectation for how these populations are also going to move northwards in the leading edge, and a large continuous range in the core. So if we think about the type of populations we might expect to see in these different parts of the range, we would expect a dominance of stress tolerance, resistance traits, and the southern edge, where populations are being forced northwards through climate change, and perhaps different traits in the core range. And whilst we can think about natural processes here, we can also think about what might have gone on with some species with importance for timber and breeding in the past, which may have affected their genetic traits in addition to the natural genetic traits that they might have within the core range. So we have different factors to focus on when we try to select which populations are interesting for us to look at for their ecophysiology in the future. So we choose to work in a provenance trial, and maybe we want to try to understand how much of this variation in phenotype is due to local adaptation and how much is due to phenotypic plasticity. And so we need to try to choose where is a good location for our provenance trial that will allow the trees that we're looking at to really express those differences related to their different origin that they have. Is it okay to look somewhere which is relatively benign for the plants to grow, or do we need to look somewhere where they're under more stress? This is a photo of a beech provenance trial that we have from the COST E52 series in the south of England at the National Broadleaf Trials, the Forestry Commission. And here the trees are growing relatively similarly, I would say, from going there to measure them. Is it a good site to have a provenance trial? This is something for us to think about in terms of whether the trees are expressing these differences that they have in their genotypes well or not. So we can look in controlled conditions instead. Maybe in a controlled environment, we can create the conditions to really see differences between plants. We have a picture here of a growth chamber where we're looking at phenological differences in bud burst in tree species by controlling the amount of far red radiation they have. But standard growth conditions have problems associated with them as well. So even if we can control both the light environment and perhaps everything, for instance, in these Aralab chambers here, controlling the CO2, the temperature, everything about the environment, then we may not necessarily see the same effects indoors in controlled conditions that we see when we look at these plants growing outside. So what is the main reason for this? Perhaps our conditions are too benign in growth environments. So here we have an example from a student of mine, David Israel, who was trying to look at aquaporin function. So aquaporins, these transmembrane proteins that serve as water channels, sometimes CO2 channels in roots, are often attributed a lot of properties when we can't explain what is going on with drought stress tolerance in plant species. But when we look at knockout mutants of these transmembrane proteins, we often don't see differences between them in benign conditions. So on this graph at the top, we have some examples of benign conditions in the greenhouse, where we grew single mutants of aquaporins and even double and triple mutants where they lost the function of these proteins, seeing very little difference between them, both in dry weight, in photosynthesis, in transpiration, in all of the factors that we looked at. However, when we put these under mild stress, so here with hydrogen peroxide, we started to see greater segregation amongst the different genotypes that we looked at. We did something similar, changing the growth conditions, just from a fully watered a greenhouse to a greenhouse where we increased the vapor pressure deficit to put the shoots under a mild drought stress. And there we saw greater segregation between these populations. So this is something for us to think about. What about when we try to do more sophisticated experiments in greenhouses? So here on the right, we have the phenotyping system at the University of Helsinki, where we have highly controlled conditions, and we can implement, for instance, here an experiment with different light filters to try to see the effect of changing the light environment on trees to see how they'll grow. But again, we have a fully controlled situation for temperature and for light. And maybe we don't see the effects that we might expect in such an environment. So we should come back to have a look at this and see what we can do to try to make realistic light environments for plants. Going back to our provenance trials, we need to try to select good locations and good provenances to look at. So this is a map showing the COST E52 trial sites for European beech superimposed over the beech distribution. So two series in green planted in 1998 and in blue planted in maybe 1995. But you can see that most of these provenances and trial sites are within the range of the species. In blue is the trial sites, and in green the provenances. And if we really want to see what the response of particular provenances might be under climate change, we need to start thinking about making these trial sites outside the species range, where they might be subject to more stresses than they would be expected to find within the range. So to this end, we planted a trial site in Helsinki. Way up here, you can see the red dot in Finland where our trial site is. And then we needed to select some provenances to go within this trial site. So we tried to look at the most extreme possibilities. So we took a provenance from southern Sweden. We took a provenance from Montejo de la Sierra in Madrid. And we took two provenances given to us by Gerhard Huber from the high Alps in southern Germany and from the Central Plains in Germany. So we expected these to be as different as we could find. And clearly we know that European beech has very high phenotypic plasticity. So we wanted to see if we could also find some interesting local adaptation. Then we need to think about why beech isn't naturally growing in Helsinki. Has it simply not managed to reach Helsinki? Or is there a reason apart from this, for instance, abiotic stress, as to why Helsinki is outside its historical range? Can it be that the soil is too heavy? Is it related to the length of the winter, the amount of daylight that the plants receive in the middle of winter? Many freeze-thaw cycles during the spring, or something affecting the timing of phenology. So this is also an important question to consider when choosing these provenance trial sites in the north of Finland. In terms of phenology, we know that this is one of the key traits for allowing beech to be adapted to the site it grows at. In 2018, this natural forest in Montejo, where we selected one of our provenances from, was completely defoliated by a spring frost in May, and we saw it only recover later in June. So with a second flush of leaves, which clearly were not as effective as the first flush of leaves. And if we see repeated extreme frost events like this in the spring, we can expect a severe decline in the fitness of these forests. So before we planted out our provenance trial site, we did some experiments on the seedlings of these 4 provenances in Helsinki, seeing how they would grow under treatments of drought, under sun and shade. And we saw that there were some differences amongst the provenances, as we'd expect, but not very great differences. For instance, those provenances growing from high elevation, showing a greater discrepancy between their growth under sun and shade. It seems requiring perhaps higher radiation rather than being so shade tolerant as some of the other species provenances. Okay, so we planted these plants out into the provenance trial. And we're able to do a series of trait measurements over time with them. And these sort of trait measurements, particularly looking at mortality, looking at phenology, looking at growth, we can feed into species distribution models like these delta trait species distribution models that we made with Marta Benito Garzón to identify the amount of variability attributable to phenotypic plasticity and the amount of variability attributable to local adaptation amongst these species, and then to recreate with these species distribution models, which I cannot get to come up on my screen. Okay, so I cannot show you them. I will just have to tell you. So when you incorporate local adaptation and phenotypic plasticity, as you probably realize, you have a more conservative effect of future changes in climate on this. So you see a smaller potential shift in the range in the future because of the amount of plasticity and local adaptation that you have shown these models. Okay, but phenology, mortality, and growth are relatively basic traits to look at. And really, as ecophysiologists, we want to try to get at the mechanisms behind these traits. So what we're able to do was to put together a large consortium of researchers from the University of Helsinki who were able to do a comprehensive campaign of trait measurements through the summer, looking not only at basic traits like phenology, but installing sap flow systems into the different provenances in this trial, looking at photosynthesis in the canopy and in the understory, comparing sun and shade, and labeling the canopy with carbon isotopes to follow the fate of the carbon through the tree, also measuring expression of genes in the leaves, in the stem, and in the roots, to really obtain a comprehensive idea of how these trees were growing under climate change. So if we can put together these different ecophysiological responses, we can then really identify where the effect is within the tree species across the different provenances, and understand what's driving some of the changes that we see. And in the end, that's what we want to identify. Okay, so I cannot get my slides to show correctly. So, okay, I will just talk to you. So, in our research group in particular, looking at the specifics, we're interested in sunlight. So why is sunlight interesting in the context of climate change? Because we know that temperature is increasing. We know that plants are moving northwards. We know there is more CO2, potential changes in rainfall, but sunlight, maybe it is not changing so much. But if we think about the actual movement of plants to different environments, they are moving polewards where the day length is longer and where there's potentially a change in the spectral composition of the radiation that they receive as well. So with respect to the spectral composition, we can think about, for instance, the environment in Finland. Colleagues at the University of Eastern Finland, Antti Tenkönen, did very interesting experiments with provenance trials where they moved silver birch provenances northwards within the Arctic Circle, where they would experience a continuous day, and test whether these northern provenances from this part of Finland were more adapted than southern provenances to this change in day length. And they identified a suite of traits in the leaves showing that there was a real difference amongst plants that had adapted to continuous day length compared with plants that were used to having nighttime within the summer. You could call this a polar day syndrome. So we know that as species move further northwards, there will be an effect of this change in light that they have. Then we can think about potential changes in the atmosphere related to how we try to address climate change. And at the moment, scenarios of climate intervention are being discussed, but may still be difficult to implement practically and without the political will to do this. Nevertheless, it should be something that we should perhaps worry about, I would say, in terms of political strategies, and get a better understanding of, so that when these ideas are discussed as plant biologists, as researchers with an interest in ecosystems and the environment, we're able to give an informed opinion of what the effects of such approaches would be. So here we're talking about things like sulfite injection into the stratosphere to block sunlight and change the environment, allowing cooler temperatures. The current strategy spoken about, for instance, by NASA and NOAA, is what's called peak shaving strategy, where rather than trying to reverse climate change, they talk about injecting some sulfites into the atmosphere to stop this from getting any worse. There are many potential problems with this, logistical problems in terms of getting the sulfites up to the atmosphere, but I'm more concerned about the ecological problems. So what will this decrease in solar radiation do? And what happens to these sulfites eventually when they fall back to Earth? So these are things to think about and to discuss, but in terms of our research into the effects of light on plants, we think about whether a change in the amount of light and the diffusivity of light is important. And when we think about diffusivity of light, we can also think about changes in cloudiness, which is something that will happen anyway with climate change, as we have changes in patterns of the weather and changes in the amount of humidity in the atmosphere, and changes in pollution. So here on the right, we have a picture from Cumbria of the forest, and on the left from Ostrava. This valley between the Czech Republic and Poland has historically been very polluted from coal mines. And in many parts of the world, as we see an alleviation of this pollution, we'll see a change in the amount of diffuse radiation received. Okay, so if we think about diffuse radiation, we know that it affects the canopy light use efficiency. And again, I'm not going to be able to show this because of how the presentation is displaying. Still don't quite understand why it's not showing. Okay. Okay. What do you see now? Do you see some animation on the screen?
Yes.
Okay, good. All right. So clearly, when you have photosynthesis at just the leaf level, this saturates at a relatively low photosynthetically active radiation. When you look at the shoot level, because of differences in the orientation and in the positioning of needles on the shoot, then you have saturation of photosynthetically active radiation at slightly higher level. And when you have a measurement of photosynthesis at the whole canopy, because of partitioning of light between the different levels of the canopy, you have saturation at a higher level of photosynthetically active radiation again. And why do you have this? Well, at the leaf level, you have photoinhibition and saturation. At the shoot level, you have changes in transmittance coming through the canopy and multiple instance angles of light on the leaf. And at the canopy level, you have this sharing across layers. And this is the classical idea we know from Paul Jarvis's research, even back in the 1980s. But when we look at diffuse radiation, then we see this increase in light use efficiency. And we need to work out what mechanisms are behind this increase. And here you can see nicely in the picture that you have big changes in light in the canopy when you have sunflex, for instance, affecting not only the amount of radiation, but the temperature and also the humidity. So we need to try to understand what is driving these changes when we see a change in light levels. Is it reduced photoinhibition? Is it deeper penetration of sunlight into the understory? Or is it only due to a change in the efficiency of physiological processes under a stable condition compared with under a fluctuating condition? So first of all, how does diffuse radiation penetrate into the understory? If we compare these two images on the left and on the right, on a clear day and on a cloudy day, we would expect on a clear day there to be high irradiance at the top of the canopy and some penetration of light through the canopy. And we can see this if we look across the images. But on the cloudy day, even though there's only a tiny fraction of that total amount of radiation at the top of the canopy, once we see penetration of light through the canopy, it's disproportionately high compared with on the sunny day, so that when we look at the amount of canopy photosynthesis in the third panel on the right, we see that this is still relatively high, even though the amount of light at the very top of the canopy was quite low. So we already understand a little bit that the diffuse fraction of radiation is having quite a large effect. If we consider a simple schematic to represent the difference between a cloudy day and a sunny day, we can see that as the diffuse fraction increases, the light use efficiency increases up to a certain point. And then once the total amount of light becomes quite low and it becomes cloudy, we see this decline in carbon uptake and decline in the total amount of light, whilst the diffuse fraction gets still higher. And we kind of understand now that the total amount of diffuse radiation is perhaps the best correlation with this. So we can look at the whole ecosystem level, combining data from eddy flux towers and from the forest floor and branches to try to integrate where the mechanism is behind these changes in radiation that we see and changes in light use efficiency. And we can do this by monitoring continuously photosynthesis and net ecosystem exchange across multiple years and seasons, and having a look to see what happens at the shoot level and what happens at the understory level. But even these sort of exercises are not ideal. You can see here that the shoots of the silver birch are being held flat in the gas exchange chamber rather than moving in the natural way that they would do if they were in the wind in the environment. So with diffuse radiation, we start to understand a little bit why we see an increase in total radiation as this penetrates through the canopy. If we look at sunflecks in the canopy as a contrast to this, then we need to think about the rate at which light changes. And these sort of fluctuations in light between different types of forest canopy can differ. So here we see a map of the length of time that sunflakes last for. And this is something that is only now possible to measure because we have very fast spectrometers that can measure at the scale of a 10th or 100th of a second. So we get to understand how light flecks make the light environment dynamic in the sun. And clearly there's a loss of efficiency in the leaves to switch on and off their photosynthetic processes at this very high speed, both in terms of the readiness of photosynthetic enzymes for the change in light environment and the effects on stomatal conductance and on assimilation. So here again, over time, in minutes, we see an almost immediate change in photosynthesis when we switch on the bright light and when we switch off the bright light, but a lag in the stomatal conductance. And so because of these changes, we know there is a reduced efficiency in high irradiance. And this is something we're able to attribute to a change in physiological processes and in morphological processes within the leaf. So what we've seen on average is if we look at the average irradiance received between a fluctuating environment and a steady light environment, we see that leaves are tending towards the morphology of sun leaves, even when they're receiving the same amount of light that's fluctuating as they would have received in a steady light environment. So this is an interesting finding and might explain a little bit why we have less efficiency in fluctuating than in steady light. But still, we don't know whether this is a long-term process or a short-term process. So in the last couple of minutes, I'll just point out a few technologies that can be interesting for us to consider when thinking about how to do this sort of ecophysiology and phenotyping more rapidly and more precisely. So I already spoke to you about how we can measure at this 100th of a second, 10th of a second level to see the variation in light that's received. You can see here in these comparisons of different stands with different types of leaf, how we see greater variation across a very short period of time in the spectra for the light environment. We also can take measurements using drones, both of leaf area index and of light within the canopy. So with my colleagues from Helsinki, John Atherton and Albert Porker, we published a nice method using a very cheap drone with a spherical camera to monitor leaf area index vertically through the canopy profile, giving very nice results if we compare an image on the left produced using this vertical drone images compared with a classical image using digital hemispherical photography, where it's always difficult to obtain a sharp image right from the canopy bottom to the canopy top. And looking across images with drones of vertical profiles, so taking measurements with upward facing sensors on drones at different levels of the canopy to obtain a vertical profile of the light going from the top to the bottom. So this, again, we can couple with leaf level measurements to understand photosynthesis across different heights within the canopy. And we obtain graphs like these ones, showing how the leaf absorption of radiation changes that we can then match with the drone flights going up and down through the canopy, and then see whether we can attribute changes in the spectral composition and amount of radiation, largely to the leaf properties or largely to the whole canopy level traits. And this is a good comparison to do. Okay, so this is a general overview with technologies and light, working together both with my team in Helsinki, collaborators across Europe and North America, and collaborators within the University of Helsinki. I think the take-home messages that I would give from this research would be that now we're starting to get gas exchange technologies, large-scale monitoring, thermal images, LiDAR, solar-induced fluorescence that allow us to look at the whole canopy level quite quickly in provenance trials and outdoors to try and understand how phenotypes are changing in different populations. Sometimes we see a mismatch between what's going on in controlled conditions and what's going on outdoors. And this mismatch may be partly due to the amount of variability within the environment, even at very short time scales. So both in light, in temperature, and in humidity, there are very fast changes in natural environments, which we don't necessarily see when things are grown indoors. Yeah, and if we can get large consortiums with different complementary sets of skills measuring different ecophysiological traits within the canopy, then we can really start to get at these mechanisms and understand the trade-offs that go on between productivity and resilience to stress in plant canopies. Okay, so I think I was going a little bit too long because of suffering with the presentation technology a little bit, but perhaps you have some questions for me.
Thank you very much, Matthew. Very, very nice talk. I'd say very, very clear and very interesting and provoking. I have a general comment, which is not about your science, but what you said about the possibility of engineering the atmosphere. Of course, I think this is, can I say it explicitly? I think this is a very dumb idea, and we could as well just wait for a supermassive volcanic eruption that would blanket the whole atmosphere. It would be fine, but this is very stupid, I think.
Yes, I agree. But it's also good to make people aware that governments discuss these ideas.
Yes, absolutely.
So that we can be well prepared as ecologists and scientists with strong arguments as to why they are not very sensible.
Yeah, I agree. Other questions for Matthew? Yes, Nico, please.
Yes, thank you. Thanks a lot. It was really nice to see this in particular because in the project in the FORGENIUS project we completely neglected traits related to, not completely, but we neglected a lot traits related to productivity and light interception. I mean, we have some remote sensing data. We do some hemispherical photographs, but we do not have gas exchange measurements. And so it's difficult to, unless by working like with soft traits such as nitrogen content or SLA, we cannot very well capture the trade-offs between resilience and productivity. So I guess, but I still miss something, and maybe you have an opinion on this, on how to estimate gas exchange. What we need in saturated condition, like the kind of water use efficiency estimates, when you have to phenotypes multiple forests in the wild, and if you have an opinion on this, probably it could be very useful for the next generation of phenotyping.
Yeah, I think it's quite complicated. Of course, we are doing things like rapid light curves at the small scale, but really, if you want to phenotype across the large scale, you want some sort of optical measurement which can capture the whole forest at once. I think there's good prospects with solar-induced fluorescence to look at these widespread changes, and people have been investigating this, even doing some fluorescence measurements at nighttime to try and capture whether there is some damage to the canopy without any noise from the daily changes in radiation that are received. So this can be quite a shrewd way to look at things. But otherwise, it can be difficult. It can be difficult to achieve. You saw that in central Finland, we have these research forests with many chambers on leaves and on the ground, but still you cannot capture the heterogeneity of the whole forest. It's too labor intensive like this.
Thanks. We have a question from Desanka. Hello, good to see you. Please.
Hi. Thank you, Matthew. Super cool presentation. Many nice and fancy things. My question is more in a more simple direction. I was just wondering, from these provenance trials in Helsinki, maybe I just simply missed it. Could you elucidate some between phenotypic plasticity? Was there some indication of local adaptation?
Sure. I did not speak too much, but I think we found that, for instance, when you had a very high light environment, those provenances that had come from a higher light environment, so from high elevation and from the south of Europe, were expressing differences in their traits. But actually, in general, we saw a lot more plasticity than local adaptation come through in our results, even though the provenances were from such different parts of Europe. Now, one of the reasons for this is because often you have local microclimatic environments which are perhaps less distinct than you would expect across different parts of the range. But again, really, when we implemented stress in artificial condition, we did not necessarily see the same response as to when we measured the plants growing outside in the provenance trial together. Certainly, the Spanish provenance was doing worse at our site, and those provenances from Sweden and from central Germany were growing better and had higher photosynthetic rate, but the differences were not as pronounced as we expected.
Okay, thank you.
Thanks a lot. Any further questions? I don't see any raised hands. Right. Well, if there are no further questions, I just wanted to the next webinar. I'm not sure I remember the date. Eduardo, do we have any information on this, or are we going to have a seminar embedded within our annual meeting? Well, we'll see. Well, in any case, stay tuned, because there will be, of course, the next webinars organized by FORGENIUS will carry on until the next summer, and then we'll probably start again a new cycle. Well, with this, if there's no further comments or questions, I really want to warmly thank Matthew for coming and giving us his view, and to Nassim for presenting the updates on the project results. And thank you everyone for attending and for engaging in the discussion. Thanks a lot.
Thank you.
Bye.